Welcome back to Human Evidently. Today we're going to talk about Paranthropus, a genus you may have heard referred to as Nutcracker Man for its notorious jaw structure. In this presentation, I will cover the three widely known species within this australopithecine genus, their chronology, anatomy, diet, and phylogeny as we understand them today. Ideally, this will help you study for any exams about Paranthropus, or just give you more information about an exciting extinct fossil hominin. Paranthropus is a robust australopith genus that occupies a central position in debates over hominin diversity during the Plio-Pleistocene. The genus is most often recognized through craniodental features associated with powerful mastication, enlarged premolars and molars, reduced anterior dentition, thick mandibular corpora, broad zygomatic arches, and frequent cranial cresting for enlarged chewing musculature. These features give Paranthropus a distinctive anatomical profile, although their interpretation requires caution. Chewing anatomy preserves functional signals, developmental patterning, and phylogenetic inheritance at the same time. The geographic distribution spans southern and eastern Africa. South African material is primarily associated with Paranthropus robustus, while Eastern African material includes Paranthropus aethiopicus and Paranthropus boisei. This distribution places robust australopiths across major fossil-bearing landscapes, including cave systems in South Africa and rift-associated deposits in East Africa. Site context therefore shapes how we interpret each species. Cave accumulations, lake margin deposits, and fluvial contexts preserve fossils through different taphonomic pathways. Paranthropus also resists any model of hominin evolution arranged as a single linear sequence. These taxa overlapped with early Homo and persisted for substantial periods. Their presence demonstrates that early hominin evolution involved multiple contemporaneous lineages with different anatomical and ecological configurations. Robust australopiths were successful participants in Pleistocene ecosystems, even though their lineage eventually disappears from the known fossil record before the Middle Pleistocene. The genus Paranthropus is represented by three widely recognized species: Paranthropus robustus, Paranthropus boisei, and Paranthropus aethiopicus. These species share a robust masticatory complex, yet they differ in chronology, geography, degree of anatomical specialization, and dietary signal. The genus ranges roughly from about 3.0 million years ago to under 1.0 million years ago, with Paranthropus robustus in southern Africa and Paranthropus aethiopicus and Paranthropus boisei in eastern Africa. The craniodental pattern is the most recognizable diagnostic cluster. Large molars and premolars increase occlusal surface area. Small incisors and canines shift the emphasis of the dentition toward posterior processing. Thick mandibles and expanded zygomatics reflect the structural demands of repeated chewing loads. Sagittal and nuchal crests, when present, indicate enlarged muscle attachment areas, particularly for the temporalis and posterior cranial musculature. This guy is considered a robust australopith. In this context, robusticity primarily refers to the chewing apparatus. It does not automatically imply a uniformly massive skeleton. Postcranial material attributed to robust australopiths generally indicates habitual bipedality, while also preserving features compatible with climbing or powerful upper limb use. This pattern fits the broader australopith condition, in which terrestrial bipedality coexists with retained arboreal competence. The genus also raises a phylogenetic problem. Shared chewing-related traits may provide evidence of close relationship, but the question is whether that evidence is developmentally and functionally linked. When several traits respond together to similar biomechanical pressures, they can produce strong anatomical similarity without resolving every branch of the evolutionary tree. Basically, the question is, are all of these species of Paranthropus related? All species of Paranthropus demonstrate a recognizable anatomical pattern, but we're not yet sure how to answer that phylogenetic question. Paranthropus robustus is the South African robust australopith. Major fossil localities include Kromdraai, Swartkrans, Drimolen, and Cooper's Cave. The species appears by about 2.3 million years ago and persists until approximately 1 million years ago to 800,000 years ago. This temporal range places Paranthropus robustus within a complex South African Pleistocene landscape that also includes other hominin taxa and diverse faunal communities. The craniodental anatomy of Paranthropus robustus includes enlarged posterior teeth, reduced anterior teeth, a deep mandible, expanded zygomatic regions, and variable sagittal cresting. Compared with Paranthropus boisei, the South African species is often less extreme in molar enlargement and facial breadth, though it is unmistakably nutcracker-like in its cranial, facial, dental, and maxillary morphology. Individual specimens show variation in crest size, cranial preservation, facial projection, and mandibular form in ways that can be expected as general population and sexual variation. The South African cave context is central to interpretation. Fossils from Kromdraai, Swartkrans, Drimolen, and Cooper's Cave are preserved in complex depositional environments shaped by sedimentation, carnivore activity, geological disturbance, and time averaging. As a result, behavioral interpretations require careful attention to association. For example, a fossil, a tool, and a faunal assemblage found in the same broad deposit do not always represent a single behavioral episode. Postcranially, Paranthropus robustus appears broadly australopith-like. Hip and knee anatomy support habitual bipedality, while upper limb anatomy suggests climbing remained part of the behavioral repertoire. This combination is consistent with a hominin adapted to terrestrial locomotion while retaining anatomical capacities relevant to arboreal movement. Claims about tool use are plausible in some South African contexts, especially given hand anatomy and associated bone or stone tools, but direct attribution remains cautious. The most defensible characterization is that Paranthropus robustus combined robust chewing anatomy, habitual bipedal locomotion, retained climbing-related anatomy, and possible tool-related behavior. The most important identifying pattern for Paranthropus robustus combines South African geography, early Pleistocene chronology, robust craniodental anatomy, and australopith-like postcranial morphology. The species appears by about 2.3 million years ago and continues until roughly 1.0 to 0.8 million years ago. Its major sites, Kromdraai, Swartkrans, Drimolen, and Cooper's Cave, anchor the South African robust australopith record. Anatomically, Paranthropus robustus is defined most securely by the masticatory apparatus. The posterior dentition is enlarged, the anterior dentition is reduced, and the mandibular and facial structures support high chewing loads. Sagittal crests occur, especially in individuals with larger chewing musculature, but crests should be evaluated alongside the entire craniofacial complex. Dietary interpretation has become more nuanced over time. Robust craniofacial morphology indicates capacity for mechanically demanding chewing, but dietary reconstruction requires additional evidence. Dental microwear, isotopic data, and comparative morphology suggest that Paranthropus robustus had a broader and more flexible diet than older research implied. Mechanically resistant foods may have served as regular dietary items, fallback resources, or seasonally important components. The postcranial evidence reinforces the mosaic character of the species. Lower limb and pelvic features are compatible with habitual bipedality. Upper limb anatomy indicates that climbing or powerful forelimb activity retained behavioral relevance. This pattern does not diminish bipedality. It demonstrates that early hominin locomotor systems often combine terrestrial and arboreal capacities. Tool use remains a valuable but cautious topic. Bone tools and stone tools occur in South African contexts where Paranthropus robustus is present. Hand anatomy may support manipulative ability. Attribution depends on context, association, and comparative probability. At minimum, Paranthropus robustus should be included in discussions of tool-related behavior. Paranthropus boisei is the East African robust australopith most strongly associated with extreme masticatory specialization. The species ranges from approximately 2.5 or 2.3 million years ago to about 1.34 million years ago. It is especially well represented in the Turkana Basin, and the holotype, OH5, comes from Olduvai Gorge. OH5 remains one of the most visually recognizable hominin crania in paleoanthropology. The older nickname, Nutcracker Man, arose from the dramatic craniofacial and dental morphology of OH5. The skull displays a broad face, large postcanine teeth, reduced anterior teeth, robust mandibular and maxillary architecture, and strong cresting for chewing musculature. The most important interpretive development for Paranthropus boisei comes from dietary evidence. Carbon isotope analyses show a strong C4 signal, indicating substantial reliance on foods such as grasses, sedges, or resources from C4-dominated environments. This evidence complicates older assumptions based primarily on cranial form. The morphology indicates chewing capacity and loading patterns. Isotopic data provide a more direct signal of consumed resources. Paranthropus boisei also demonstrates the value of sample size. The East African record includes multiple cranial and mandibular specimens, allowing researchers to compare variation across individuals and sites. This broader sample shows the consistency of the robust masticatory complex, while also revealing variation in preservation, age, sex, and regional context. The species overlaps temporally with early Homo. That overlap can help researchers partially reconstruct early Pleistocene ecosystems. Paranthropus boisei existed alongside other hominins, stone tool traditions, carnivores, and changing habitats. The species was likely part of a diverse hominin community in its region, filling a specific niche among other hominins. The concise diagnostic profile for Paranthropus boisei includes East African distribution, early Pleistocene chronology, the largest molars among known hominins, massive jaws, and a strong C4 dietary signal. OH5 from Olduvai Gorge is the holotype, while the Turkana Basin provides an especially important comparative sample. The dentognathic system of Paranthropus boisei is among the most specialized in the hominin record. Molar and premolar enlargement increase processing area. Reduced incisors and canines emphasize posterior chewing. The broad face and zygomatic expansion support enlarged chewing musculature. Cranial cresting indicates muscle attachment demands. Together, these features identify a masticatory complex capable of substantial repetitive loading. The C4 dietary evidence requires careful interpretation. C4 signals may reflect grasses, sedges, underground storage organs from C4 habitats, or other resources connected to open or wetland environments. Paranthropus boisei appears to have relied heavily on resources distinct from those emphasized in many other hominin dietary reconstructions. Regarding tool use, some evidence suggests Paranthropus boisei lived in contexts where stone tools were present, and some postcranial material has been interpreted as compatible with climbing-related or powerful upper limb behavior. Direct attribution of tool manufacture or use remains challenging. The presence of Homo in overlapping contexts complicates the assignment of tool use to Paranthropus specifically. For identification, the most reliable pattern is the combination of East African provenance, extreme postcanine megadontia, massive jaws, broad face, and C4-focused dietary evidence. In comparison with Paranthropus robustus, Paranthropus boisei tends toward greater masticatory exaggeration. In comparison with Paranthropus aethiopicus, it is generally more derived in facial architecture and less prognathic. Paranthropus aethiopicus is represented most famously by KNM-WT 17,000, the black skull from West Turkana. This species is central to discussions of the early East African robust lineage. Its importance lies in the combination of robust masticatory traits with cranial features that appear more primitive than those of later Paranthropus boisei. Chronologically, Paranthropus aethiopicus is placed roughly between 2.8 and 2.1 million years ago, primarily in Ethiopia and Kenya. This range places it near the emergence and early diversification of robust australopiths. During this interval, the hominin record includes multiple taxa with different anatomical configurations, and early Homo is also becoming relevant to broader evolutionary debates. KNM-WT 17,000 shows large posterior teeth, strong cranial cresting, a small brain, and a more prognathic, sloping face than Paranthropus boisei. The specimen shows that parts of the robust masticatory complex were already developing before the later extreme morphology seen in Paranthropus boisei. The fossil record for Paranthropus aethiopicus is fragmentary. Researchers often treat Paranthropus aethiopicus as close to the ancestry of later Paranthropus, especially Paranthropus boisei, but the precise relationships remain open to debate. This is a normal feature of paleoanthropological inference. Fossil taxa are often built from incomplete samples, and phylogenetic hypotheses change as new material, new dates, and new methods become available. For identification, focus on the black skull profile. Early East African robust australopith, large posterior teeth, cresting, small brain, strong prognathism, and a sloping face. Those features distinguish it from the later and more facially derived Paranthropus boisei. The key interpretive role of Paranthropus aethiopicus is its position near the base of the East African robust lineage. With a range of about 2.8 to 2.1 million years ago, it appears earlier than much of the classic Paranthropus boisei sample, and is commonly discussed as an ancestral or near-ancestral form within Eastern African Paranthropus. The black skull exhibits large posterior teeth and robust cranial features, but it also retains a smaller neurocranium and a more prognathic, sloping face compared with Paranthropus boisei. Ethiopicus shows robust masticatory traits that appear alongside more primitive craniofacial features. The fragmentary nature of the record makes Paranthropus aethiopicus a taxonomic challenge. A small fossil sample limits the ability to assess variation within the species. It also complicates comparisons with Australopithecus, early Homo, and later Paranthropus. Diet is also less securely reconstructed than in Paranthropus boisei. The species appears less isotopically and anatomically extreme than later Paranthropus boisei, though the available evidence is also more limited. It may represent a broader or earlier dietary ecology within the East African robust lineage. The species can be summarized in 5 linked terms: early, East African, black skull, robust chewing anatomy, and primitive craniofacial retention. For research-level discussion, the species is valuable because it exposes the difficulty of distinguishing ancestor-descendant relationships from shared functional morphology in a sparse fossil record. A comparison of Paranthropus robustus, Paranthropus boisei, and Paranthropus aethiopicus should begin with geography and chronology. Paranthropus robustus is the South African species, known from sites such as Kromdraai, Swartkrans, Drimolen, and Cooper's Cave. Paranthropus aethiopicus and Paranthropus boisei are Eastern African species, with Paranthropus aethiopicus appearing earlier and Paranthropus boisei continuing later into the early Pleistocene. Phylogenetically, Paranthropus robustus and Paranthropus boisei are often treated as sister species, while Paranthropus aethiopicus is commonly viewed as an earlier branch or possible ancestor near the base of the eastern robust lineage. This arrangement is plausible, but it is not immune to revision. Shared masticatory traits can be phylogenetically informative, yet they may also evolve together under similar functional pressures. Diet provides one of the clearest contrasts. Paranthropus boisei shows the strongest C4-focused dietary signal. Paranthropus robustus appears to have had a broader and more flexible diet. Paranthropus aethiopicus is more difficult to reconstruct, though it may have been less specialized than Paranthropus boisei. All three species overlapped with Homo in at least some temporal and geographic contexts. This overlap is central to understanding early Pleistocene hominin diversity. Homo and Paranthropus represent different adaptive configurations within the same broad evolutionary landscape. Their coexistence complicates behavioral attribution, especially when tools and fossils occur in the same deposits. The reason for their extinction remains unresolved. The lineage disappears by under 1 million years ago, but no single cause has achieved consensus. Climatic instability, habitat restructuring, dietary specialization, demographic vulnerability, competition, and sampling gaps all remain plausible contributors. Extinction is most likely a multi-causal problem, rather than a single event explained by one variable. The three species of Paranthropus share a recognizable, robust masticatory pattern, yet their differences are central to interpretation. When traits evolve as an integrated complex, they can make taxa appear more closely related than they are. This is why robust australopith relationships require evaluation through multiple lines of evidence. Dietary reconstruction provides a second level of complexity. Paranthropus boisei has the strongest C4 signal. Paranthropus robustus appears more flexible. Paranthropus aethiopicus remains more uncertain, although it seems less extreme than later Paranthropus boisei. These differences are essential for avoiding overly broad claims about the Paranthropus diet. The genus also demonstrates that hominin diversity during the Plio-Pleistocene was broad and regionally structured. Robust australopiths coexisted with Homo and persisted for long periods. Their eventual disappearance should be analyzed through ecological, demographic, and climatic frameworks, rather than through assumptions about evolutionary inferiority. So, boiled down, Paranthropus is a long-lived, geographically structured, morphologically specialized hominin genus. Its species preserve evidence of powerful chewing adaptations, dietary diversity, locomotor mosaics, and phylogenetic ambiguity. A strong interpretation of the genus requires the integration of comparative anatomy, stratigraphy, geochronology, dietary chemistry, tool context analysis, and evolutionary theory. Special thanks to my Patreon subscribers, Red Fox the Starfish, Nori the Salty Cat, and Gary, Jack, and Mark the Porpoises. The value of your kind support is immeasurable. Thank you for spending this time with me on human, evidently. I hope you're leaving with a clearer understanding of the subject and a deeper curiosity about the story of life, evolution, and our place within it. Until next time, keep asking those good questions. If you want to support my channel, please consider subscribing here and on Patreon, where you can access my content for free or contribute a small donation to get your name at the end of my videos. I am working on a series of educational material for middle grade readers and 2 science fiction novels. Such endeavors are only made possible by contributions from viewers like you.